Yes, go ahead, please.
Yes, your screen is very clear. Go ahead.
Okay, thank you. So, hello everyone. So I must say that I only have like 5 minutes for this presentation. So kindly stay with me while I go through the slides. Okay, so welcome once again to the project presentation on phylogenetic analysis and evolutionary dynamics of Pseudomonas aeruginosa isolates from diverse geographical region. This presentation is brought to you by the Cohort 1 of the Genomac Internship Program, and my name is Ahmed Norain Kinde, and I am joined by my project team members, Wahab Yusuf Adeoye Marvelous, Aziz Dauda, Nika Amakom, and then Moidin Arugundade. So today's presentation, we cover the following outlines. So starting from the introduction, so Pseudomonas aeruginosa is a ubiquitous rod-shaped aerobic non-spore-forming bacilli with a single flagellum for motility. This bacterium is known for its environmental ubiquity and high antibiotic resistance, making it a major cause of nosocomial infections, which are infections acquired in the hospital. So illness caused by Pseudomonas aeruginosa include osteomyelitis, which is the infection in the bone. We have pneumonia, we have bacteremia, which is the presence of bacteria in the blood. We have eye and ear infections, central nervous infections, and others, as you can see in the image on the screen. So our primary objective was to analyze the genetic diversity of various Pseudomonas aeruginosa strains by comparing their 16S rRNA sequences, sorry. Also to determine the evolutionary relationship among the selected strain using phylogenetic tree, and lastly to evaluate how geographical origin and environmental sources contribute to the genetic variation observed among the selected strains. So why are we using 16S RNA? So 16S RNA is actually a very high, or is a highly conserved gene found in all bacteria, and is commonly served as a phylogenetic marker to identify and classify bacteria species. So for our study,
That reduce the
for our
volume.
Sir, please, your slide is not
Yes,
moving.
the
Can you
first
please change your slide? And kindly, please, everyone, mute your mic. Thank you very much. Please mute your mic.
Okay, I'm actually on
Yeah,
Marketing and
please.
Methods now. Are we on the same
Wait,
page?
wait. No, no, no. Can you present entire screen? Every other person presenting is our project. Please present entire screen. Don't present the windows, present. So stop
Okay.
presenting and present again. Thank you.
We're so sorry for the delay. Thank you very much for staying with us.
Hello, everybody. We're going to be sharing the YouTube link.
How about now? Is it better now? Hello, anyone?
Yes, the
Can you
slides
put it
are
on
changing, but please, can
slideshow?
you make it full?
Can
How
you put
about
it on
now?
Yes, thank you
slideshow?
very much.
Okay, so it's moving now, right?
Yes, it is. Please, everybody, mute your mic. Thank you very much. Please mute your mic.
Okay, so I'll just start from the introduction then. Okay, so I said for the introduction, so we're actually working on phylogenetic analysis and evolutionary dynamics of Pseudomonas aeruginosa isolates from diverse geographical locations. So, and here are the outlines. So starting from the introduction, I said earlier that Pseudomonas aeruginosa is a rod-shaped aerobic non-spore forming bacilli with a single flagellum for motility. So this bacteria is known for its environmental ubiquity and high antibiotic resistance, making it the major or a major cause of infection acquired in the hospital. So here are the infections caused by Pseudomonas aeruginosa, which includes the pneumonia, the bacteremia, the respiratory tract chronic infections, urinary tract infections, and others. So the objectives of the work is to analyze the genetic diversity of various or of selected Pseudomonas aeruginosa strains by comparing their conserved 16S rRNA sequence, and also to determine the evolutionary history or relationship among these selected strains using a phylogenetic tree. Also to evaluate how geographical origin and environmental sources of these strains contributes to the genetic variation. Okay, so I said, why did we use, or why are we using 16S rRNA gene to carry out this study? So we are using this particular gene because it's a highly conserved gene that is found in all bacteria and is commonly serves as a phylogenetic marker to identify and classify bacteria species. So for our study, we used tools and database such as NCBI GenBank for sequence data, Genius software for sequence analysis, and also about the data retrieval, we retrieved 50 16S rRNA sequences of Pseudomonas aeruginosa strains across different geographical location and sources, such as clinical, environmental, animal, and plant sources. And here is the breakdown of the country where the strains were being isolated from. So following that, we then carried out multiple sequence alignment using Mozo alignment method and its maximum number of iterations. And then we constructed phylogenetic tree using neighbor joining method with a bootstrap value of 1000. So this is the map of the study showing the geographical distribution of 50 Pseudomonas aeruginosa strains. So this visual representation helps us to understand the broad geographic coverage and diversity of the sources from which strains were isolated. So our workflow actually involves several key steps, starting from the data retrieval to obtain 16S rRNA sequences, then importing the file into Genius software. Then after that, we conducted multiple sequence alignment, which we later used to visualize the matrix heat map, and then used it to construct phylogenetic tree to understand the evolutionary relationship among the strains. So this slide shows the multiple sequence alignment of 50 Pseudomonas aeruginosa strains conducted using the Mozo alignment algorithm in Genius software. So these alignment helped us to identify the conserved and the variable region within the 16S rRNA sequences across the strains, which are crucial for phylogenetic analysis. And here we have a factual representation of the matrix heat map showing the genetic similarities and differences among the 50 strains. So the heat map indicates the percentage of the identical bases or residue among the strain, with different colors representing the highest, moderate, and lowest percentage identity. And here, this is the phylogenetic tree of 50 Pseudomonas aeruginosa strains. And the tree is actually divided into several major branches with notable cluster of strains indicating close evolutionary relationship. So we actually have two primary branches here, which is the first two here, and the larger grouping strains, making it two primary branches from the original. Okay, so these branches are supported by significant bootstrap value. As you can see here, we have 61, 51, 96, and 78 to ensure the reliability and confidence level of this grouping. So there is actually a considerable genetic diversity among this larger group. As we can see, that this first two is actually from Iraq, while the others are from Germany, India, Pakistan, China, Nigeria, and others. So the last two strains here, the last two strains here, which is the PO88UC strain and the CHA14NQ strain, are positioned on separate branch, indicating a unique genetic makeup that may result from distinct evolutionary pressure or geographical isolation. So generally, we can say that strains collected from similar sources or geographical locations tend to cluster together. For instance, several strains from India, Pakistan, and Iraq form distinct group. So our key findings for this research work is that first, the integration of phylogenetics data with geographic metadata provide valuable insights into genetic conservancy and divergence of Pseudomonas aeruginosa strains, and also the high conservancy among India strains and significant divergence among strains from other countries reflects the profound impact of regional environmental condition on the genetic evolution of this species. So for future directions, further research on a broader range of geographic locations and isolation sources is needed to enhance our understanding of Pseudomonas aeruginosa's evolution. And in conclusion, this research contributes to a deeper understanding of Pseudomonas aeruginosa's genetic diversity, evolutionary relationship, and geographic distribution. And the distinct clustering patterns highlight the impact of different environmental and clinical settings on the genetic structure of these bacteria, which is crucial for understanding the adaptability and guiding infection control and management strategy. So thank you for listening. We appreciate your time and attention. Thank you.
Thank you so much, Mr. Ahmed, for this very beautiful presentation. I mean, you can see what Genomac is doing. Genomac is doing a whole lot. Great innovations, great research are going on. Thank you so much for this beautiful presentation. We're so glad to have this. Candidates 2 should please start getting ready to come on board. And please, you strictly have 5 minutes so that we can finish up on time. 5 minutes, please try as much as possible to be time conscious. Can we have candidates 2 on board?
Hello, everyone. Good evening. I'll can you share my screen now? Okay. Please, can we all see my screen?
Yes, we can.
All right. Cohort 2, our program was targeted at metagenomics in cancer research. So the title of our program was to find the functional insight into the microbial genes associated with pancreatic cancer promotion and progression. We looked at it via a metagenomic approach, where our aim was to uncover the role that microorganisms play in the microbial genes played in promoting and advancing pancreatic cancer. And we wanted to identify the key genes, which these are our objective, identifying the key genes and the functions of these microbial genes and their role in cancer development. We wanted to also uncover how these genes help contribute to cancer progression, and also wanted to discover the potential targets biomarkers among these genes. So as we all know, metagenomics is the study of this entire nucleotide sequence that you isolate from an organism. It can be even from an environmental sample. And so our analysis took this shape. We collected our sample, we carried out, and we specifically collected the shotgun metagenomic sequencing, as it gives us it gives us the entire sequences, including, it gives us both the functions. So we're able to analyze both the taxonomic classification, we're able to get the analysis of the population, we're also able to get the functional analysis to know the functions of these organisms in the environment, in the microbiota. Then the methodology we used, we retrieved our data, we carried out the comprehensive metagenome analysis, we carried out resistance profile study, we carried out virulence profile study, we carried out the mobilome analysis, biomarker identification, and taxonomic analysis. So I'll try to shrink everything in these 5 minutes. So this slide shows the data retrieval. The data was retrieved. We got the data from a publicly available data, that's NCBI, and we're able to get out this information, the samples, the IDs, the size of the data we used, because as it's important. So we generated the data from NCBI. Slide 2 shows the comprehensive metagenome analysis. Now, the reason for this is we carried out using a BVBRC tool, we were able to get the characteristics of we're able to get the characteristics of the chromosome. This gives us insight into the length, the GC content, gives us insight into the protein content, it gives us insight into the functional annotations of the gene, as we are going to use it for further analysis. Then the next step we did was the subsystem. This subsystem also helped us to check the functional categorization. We used different tools such as the Galaxy, and we also used the BVBRC as well. The heat maps you see here, we got the subsystem functional characterization. Excuse me. So the subsystem characterization was carried out to identify specific functional characterization as it relates to the progress of the pancreatic patient, the progress of these organisms in the pancreatic cancer in a patient. And we had this A represent the PDAC patients, that's these people that already have the cancer, and the B is the control people, the non-tumor patients. So we were able to find out using the closed vid, we're able to construct a heat map to be able to find the relationship between the organisms and to check how they contribute to the progress of the pancreatic cancer in patients. The mobilome analysis. The mobilome analysis helped us to, the heat map here revealed the distinct patterns of virulence gene abundance. So what this analysis helped us to check out the abundance of the gene. So entirely in the whole research, what we wanted to check, what is the prevalence of these genes in the tumor patients and non-tumor patients. And with this heat map, it helped us to analyze the patterns of the virulence gene, the abundance of the gene, which gave us insight into the mechanism of the pancreatic progression. So the PDAC, if you notice, according to the maps, we notice the mobilome elements that are highly prevalent, that's the transposons that are highly prevalent, unlike in the control. We also carried out the resistance profile study using the resistance profile study using the heat maps as well, and we're able to check out in the resistance profile, we wanted to check the genes that contribute. We wanted to check the genes that are prevalent in the samples, and the prevalent sample, we found out that the 5 common antimicrobial resistant genes that are prevalent, which are the DDLs, the EFG, we have many others, and it showed the presence of antimicrobial genes that are very prevalent in the oral metagenome, which could be as a result of gene transfer from the gut due to dense microbial population in the environment. So there can be a transfer from the gut to the pancreas. So that can be a result of why there is a high prevalence of these genes in the patients. Then we carried out resistance mechanism. The essence of this resistance mechanism, is to check the classifications of the organism, how they contribute, the impact of the cancer treatment, and the antibiotic classifications and how they contribute to the growth and the progression of the cancer in the system. I'm sorry, because of it's not very obvious here, but it showed that different genes, it showed different classes that were prevalent in this, and we have them as example of such organisms, sort of such classification is the efflux pump, which that confers antibiotic resistance 65% in 65% of the sample. Or we also have some classifications such as the inactivation enzyme, which was detected in 50% of the antibiotic resistant variants. We also carried out the virulence profile in the fecal bacterial virulence gene. Our studies captured the fecal and the oral speech, fecal sample and the oral sample. And here we try to check how does the virulence gene connect or relate to the bacteria that we found in the samples that we've gotten, and we're able to analyze them, and we have this, and we noticed that there were genes that were present. There were genes that were present and very dominant than the other genes. One of the dominant genes include the AR gene, ALR gene, the ARG genes, and the ARO genes, which might not really show because of this. We also have so this heat map shows the sources, and here we try to link based on our general sequence properties. We observed the sources of this organism. So here we try to look at how does these sources of this organism affect the virulence factors of these organisms, and that was what we got here. Then this Figure 7 heat map shows us the taxonomic relationship between our organisms. We carried out taxonomic analysis at the genus level and the species level on the taxonomic composition and the abundance profiles in the tumor microbial environment. We made use of cracking to compare the sequences and assign them the labels to each of the sequences. We also use this to check the abundance profile between the cancerous and the non-cancerous cells to identify the groups that are significantly abundant. And from here, we notice.
Ms. Faith, kindly round up. Your time is going overboard. Thank you very much. Kindly round up your presentation.
All right, so I'm already done. From the heat maps, you notice that there are places the organisms are much abundant than in the control. So we wanted to check which of the organisms are prevalent in this taxonomic analysis, comparing between the cancerous and non-cancerous patients. In conclusion, the study of these organisms provided insights into the mechanism of the PDAC development and progression, and it helped us to know the treatment effectiveness and overall patient prognosis. And based on studying and looking at it, the females are 60% more prevalent than in males that are 80%. The 80% prevalence in males are found in the non-tumor samples, while the 60% prevalence is found in the tumor samples. These are the few references I have. Thank you very much.
Thank you very much, Ms. Faith, for this beautiful presentation. This is great. I mean, you can see what is happening here in the world of Genomac. Genomac, like I said, is one of the top biotechnological companies that are doing a whole lot in bioinformatics and research as a whole. So yes, kindly please, you can please exit, some of you can please exit this Google Meet and join via YouTube so we can have our other presentations on board. Presenters are not presently on this call, and because of the great deeds that Genomac is doing and performing, we have over 100 people on this call. So would love some of you to join through the YouTube link that has been provided on the group so we can have our presenters on board. Thank you very much. Please kindly let our presenters join this call. And I can see a few people raising their hands. Is there a reason why? I mean, Is there a way we can help you? Mohammed, Oni, and Eneche Okuri, your hands are raised. Is there a way we can help you? Is there something you need from us? Please let us know via the chat room. Yes. Thank you very much. So now we'll be having candidates three for their presentation. Kindly mute your mic if your mics if you're not to present. Please kindly mute your microphone. Zachariah, please can you hear me? Kindly mute your microphone.
I'm sorry, I mute.
Thank you very much for muting. So we'll have the next presenter on board. Thank you. Like I said, please
Am I?
kindly join via YouTube so we can have our presenters on board. Shreya, is there a way we can help you?
Yes. Am I audible, ma'am?
Yes, you are. I can hear you clearly.
Okay. Thank you for giving the opportunity. So here I am presenting my work. It is a cohort-based study. It was funded by and it was sponsored by and organized by Genomac Hub, exploring the health enhancing and strength specific traits across the diverse probiotic strains. And under this, our probiotic research group, under the mentorship of Mr. Stephen, David, me, and Izio Piora, have successfully completed main study of aim of the study, and thankful to the CEO of Genomac Hub. So we all know the specifically the probiotics is a new it has been trending because it is fullness for the treatment of autoimmune conditions and allergic reactions, metabolic disorders. So and also as an alternatives or additives to the antibiotic treatment, there are lots of probiotics in the markets.
Sorry, Miss
And
Shreya, your slide is not moving. Please, is there something? Your slide is not moving. It's still on the first page. Please kindly
uh,
rectify that. Thank you.
am I in the second page or third page?
You're still on the first page. So
Okay,
please
okay,
kindly
sorry.
rectify that. Thank you.
Is it visible now?
Yes, it is. You can go ahead.
Yeah. So as I was saying that probiotics, it has been trending in the food industry and the research of its usefulness for the treatment of autoimmune conditions, allergic reactions as well as metabolic disorders, and it has been applied as an alternatives or additives to antibiotic treatments. In the markets, we can see lots of probiotic and its market use and the industry's use. So because and the we can say that probiotic provide a beneficial effect by modulating the host immune system by the release of the antimicrobial substances or through competitive exclusion of the pathogenic bacteria. So these are the probiotics usefulness, enhancement of the epithelial barrier, inhibition of the pathogenic bacteria. So the aim of our study was to explore and analyze the health enhancing traits present in various probiotic genome species, cross genus comparison and evolutionary relationships of Lactobacillus rhamnosus, lactis, and fermentum and longum. For today's presentation, I'm going to present all the results from the Lactobacillus rhamnosus results, and also as well as antimicrobial profiling of the selected strain, that is Lactobacillus rhamnosus. So for the to retrieve the data, we have used 25 assembled genomes of Lactobacillus rhamnosus from the NCBI databases, and then we used for the genome annotation, we use the Patrick 3.69 annotation pipeline Rust toolkit, and for the predicted open reading frame, we use the Patrick using
Miss
these
Freya,
pipelines.
sorry to cut you short. Please, can you
Yes.
please, some of you can please drop out of this Google Meet and please join via our YouTube link so we can have our presenters on board. Thank you. Please, you can leave this meeting and join via our YouTube link that is provided on your group so we can have our presenters on board. Thank you, Miss Shreya. You can go ahead. You're
Ma, I
doing a great
cannot see
job.
the link. I can't see the link. I
The
can't see the
link is in
YouTube
your
link.
group chat. In your group chat. Check your group chat for the YouTube link. Thank you. Miss Shreya, please can we have you back on board? Miss Shreya, please can we have you back on board? Are you still on this call? Then the link has also been dropped on the chat room, so you can check that. Miss Shreya, please, are you still on this call? Can we have you back with your presentation? Thank you. Please let me know. Can you kindly signify?
She just messaged on the group that she's out. She's out of the Google. She needs to rejoin the Google.
Thank you very much. I'll do that. Um,
Hello, can we have number three? Can we
um,
have number three?
dear sir, this is Miss Sharia is the third candidate, so I think maybe we should move to the next program while we wait for Miss Sharia. Okay, please, while waiting for Miss Sharia, I think she probably mistakenly left the meeting. We'll
Yes,
have
ma'am.
our
I've rejoined. I have rejoined.
okay, you're back. Thank you very much,
Yeah,
Miss
yeah.
Sharia. You have
I
this.
thought you. Sorry for the miscommunication.
No problem.
Um,
Please hurry up. You have just 5 minutes. Thank you.
Yeah. So these are the materials methods. So for the NAI analysis and the phylogenetic analysis, we use a J species W pairwise genome comparison web server and phylogenetic tree for using the Patrick tree builder. And for anti-smash we web server we use for secondary metabolite prediction and resistance profile analysis we did against the specialty gene databases using AirDB card and in the Patrick blast. So these are the results. So using the phylogenetic tree, we evolutionary relationship, we detected that Lactobacillus rhamnosus and Lactobacillus fermentum sharing the most closely relation, as we can see from the same branch from the ancestor, they have shared the same evolutionary relationship, where lactis has been the kind of the offspring of the parental of rhamnosus and fermentum, and bifidobacterium longum is the mostly distantly related to the other strains of lactobacillus bifidobacterium longum, because you can see this is a parental the ancestor, it has been evoluted from the ancestor directly. And from these heat maps we have tried to understand the interspecies in relationship of similarity, and this similarity is as the degree of from red it is high similarity, as it is reducing to black it is lesser similarity. So here we can conclude that Lactobacillus rhamnosus DMS4 as sharing high similarity with the Lactobacillus LRB1, where Lactobacillus rhamnosus BPL5 is showing less similarity with the rhamnosus LRB1, and this could be the reason because the nucleotide basis the composition, not the order. So this way, even in the interspecies, we are seeing the similarity or the dissimilarity. So moving on to the next results. In this heat map, we have tried the antibiotic class against the
Can you
resistance
share? Your
mechanism.
slide is not moving. Sorry, your slide is not moving. Put it on full screen.
Yes, sir. It is on the full screen mode only.
Please put it on full screen. At the lower
No,
part,
we
your slide is
no
not
no.
moving.
You are not on full screen. Can you? Okay, can you stop sharing and share again? But this time around, pick full screen. Share
Okay,
as
sorry,
full
sir.
screen, entire screen.
Yeah,
Yeah.
okay, sir.
Stop sharing and share again,
Uh,
but this time around as entire screen. Yeah.
Now is it visible, sir? Hello.
Yes,
Yes,
continue.
thank you
Yeah.
Yes.
very much.
Sorry for the inconvenience. I apologize. So the antibiotic class against the resistance mechanism, and we have seen that peptide antibiotics only show the high efficacy against protein altering cell wall change, conferring the antibiotic resistance as well as the regulatory of the antibiotic resistance gene, and the triclosan triclosan only show the medium efficacy, because as in the degree of red it is showing the higher amount of resistance, and lesser is green, it is showing the medium efficiency against the medium degree of resistance against the antibiotic mechanism class, and the black it is showing the lesser resistance. So all other antibiotic class show medium efficacy against the antibiotic target in susceptible species. If we come, we have studied also the sample against the gene. So all the strains of the Lactobacillus rhamnosus, Lactobacillus rhamnosus have low AMR genes, which is characteristics of the probiotics. However, the strain, particularly you can see here, the red color, it is only is seen in the strain 1.302, so has more AMR gene relative to the other strains. Moving on to the next, that the gene against the resistance mechanism. Here we can see these are the genes, and that these are the mechanism, the resistance mechanism. So the genes indicating the red functions in the corresponding resistance mechanism, while the black ones no function in corresponding resistance mechanism. And also this is a gene against the antibiotics particularly. These are the genes in the x-axis, y-axis. These are the genes, and these are the group of the antibiotics. So genes indicating the red show resistance to the corresponding. These are the antibiotics group. Sampled against the resistance mechanism in this map, we can see the resistance mechanism implied by all the strains are the protein altering cell wall, we can see, and cell wall and conferring the antibiotic resistance and regulator modulating the expression of the antibiotic resistance gene as well as the antibiotic target in susceptible species, which is only one highly employed. And a sample against the antibiotics, these are the group of the antibiotics, and these are the samples. So sample against the antibiotic class, all the strains of resistance to peptide, particularly this peptide antibiotic group, with the highest found in the strain 1.32, because the red color is, as I mentioned, it is the highest degree of expression. Black, it is a lesser degree. So also all the strains of medium resistance in case of aminoglycoside and the aminoglycoside, and cytosine, that's why it shows, it gives a green color of green color. So this is we have tried the mapping against the sample against the antibiotics. So all the strains, so high resistance to daptomycin with the highest resistance seen in the as we had already mentioned, 1.302. So after the secondary metabolite distribution across the Lactobacillus rhamnosus, we have found that the saccharide production is a common feature among these strains, and only 7 strains are capable of type 3. The blue color indicates the polyketide synthesis, and we can say there are 7 across the 7 strains, and fatty acid synthesis conserve a function. The red stands for the fatty acid synthesis, and we can see in all the strains the red color is present. The ribosomally synthesized and post-translation modified peptide is not universally present across all strains. That's why it is very lesser number. And lasopeptide production is only found in the KF7 and the FMB. So that's why you can see the green color is represent the lasopeptide. So it is present in the FMB 14 and the KF17. So, and the lanthipeptide class 4 is rare and only found in the KF7. So that's why the color, this color, the light yellow color is only present in the Lactobacillus rhamnosus KF7. So this was the result of
Miss Freya,
distribution.
please kindly round up. Thank you very much. Just kindly round up. Thank you.
Yeah, sure. So after and the fermentation capabilities across, we found all the strains as were highly effective in the, this is a mixed fermentation, and this is a lactate fermentation, while the low effectiveness in case of butanoid metabolism. And the stress response, we found this all the strains has showing uniform response, while these strains are effective in the fusidic acid resistance. These are the antibiotic resistance genes. And this is the overall overview of the pangenome graph of all the Lactobacillus rhamnosus. So this is the alignment where from there we found that 56 unique coding sequences are found, and only this strain is sharing this unique coding sequences, and the highest number of health benefit conferring CDAs, that is particularly sequencing the protein of our interest. That is the strain BPL5. These are the health benefiting CDSs present in the BPL5. These are the positions are also mentioned. So this we have found secondary metabolic profile against the
Sorry, Miss Freya, your voice is echoing. We can't hear you properly. Please kindly rectify that. We
right.
can't hear you properly.
Hello.
It's
And
not sounding great. Please, can you rectify that? We can't hear you well.
yeah, sorry.
Thank you.
And the strength, am I audible now?
Yes, you are. You can please go ahead.
So secondary metabolic profile show that the antimicrobial activity against these pathogens. So they kill if our gut microbiota, the lactobacillus rhamnosus, these strains are present as well as the pathogenic bacteria are present. Then they will kill these pathogens and then support the colonization of the good bacteria and persistence of the probiotic properties. Will help in the modulating the host immune response. And the most potential was in this regard VH probi M12 and strain BPL5 is highest number of health benefit conferring the CDS. These are the properties, these are the health benefits of the probiotic properties, and all strains were efficient in the case of carbohydrate mixed acid metabolism. That is a good sign because it will help in the gut colonization, and all the strains except 1.3020 are susceptible to different antibiotics and classes. So this we can use this conclusion in further studies as well. So the conclusion for the final conclusion of our study was the L. rhamnosus presence in the gut confers health benefit, and excessive use of antibiotic deplete the population of rhamnosus and predisposes a health risk by the virtue of resistance to the antibiotics. These are the references, and thank you so much. And sorry for the inconveniences I caused. Sorry.
Thank you so much, Ms. Freya, for this beautiful presentation based on the research that you've done. I'm sure you would agree with me now that Genomac is doing great. Genomac is doing great with the presentations that we've had via this meeting and call. I am more than surprised and astonished. I mean, thank you so much, Genomac, for this great opportunity. So now we would invite the brain behind Genomac existence, our own dear CEO, Mr. Oluwaseyi, to come and give us the orientation course for the new cohorts 3.0 that will be joining us. So please, kindly welcome Mr. Oluwaseyi on board. Thank you very much, sir. You can come on board now. Okay, sorry, before that, I can see you raising your hand. Mohammed, Precious, Zeshan, Abdurraham, and Zachariah. Is there any way we can help you? Is there something you want us to do for you?
Okay. Good
Bartholomew.
evening. Yeah, good evening. I wanted to
Good
know
evening.
if the other groups won't be presenting.
They will be presenting. Please, you'll be presenting.
All right, thank you.
Yes. Mohammed, Precious, is there any reason why you're still raising your hand? Yes, please, can hear you.
Hello. Mine is just to ask whether if you can get the video links as well as like just the slide, if you can go through, even though we don't know how they conduct the research, but we have seen their findings, their result, as well as the method. And of course, the finding of their research is actually quite impressive and interesting. I don't know how can someone join this group of Genomac. Is it open to anybody like to do a research using computational method? Because there are a lot of data as one of my sister that presented on cancer metagenomics. Of course, there are a lot of data in which someone can explore into by using CBio portal and TCGA to explore into like racial disparity among others, as well as the copy number alteration that exists in different genes, okay, such as ploidy and purification of a particular gene. So all these are some of the data which are available at our disposal for us to consider analyzing them, and of course we can study different genomic alteration that occurs in different among racial group. So I don't know whether if you can get into that particular website, see by your portal. So is it open to any body to join Genomac? Who?
Sorry, Zachariah, please. Are
All
you
right.
one of our new students, or you're graduating?
No, I just joined I
Okay,
got
okay.
the link and I
Can I,
registered, so
yeah,
I'm
can I
just
Our
come in?
following.
CEO would address that. Thank you very much.
Moderator. Okay, yeah, yeah. So let me address that. Thank you so much. So, Miss, what's your name again?
Zakaria Muhammad Bello from
Okay,
Brazil.
okay, from Brazil. Nice, nice. So, it's nice having you on board. All right, it's nice having you joining us at this graduation and orientation program. So, based on the questions you have, I can see that you have perspective to some kind of research in silico using genomics and bioinformatics. All
Yes,
right. So,
yes, yes,
and then you're talking about the database, you're talking about data sets on the public database, isn't it? Samples on the
yes.
public database like NCBI, if they can actually be used to, if they can actually be
And
utilized to carry out research
yes,
and projects and even towards publication and all of that. Yeah. So this
yes.
project just finished right now. The presentation of this project that just concluded now, the people, I mean, the groups were actually in the cohort 2 of our research internship program, which is free.
Okay.
All right.
Okay.
Exactly. So we have cohort 3. So we are doing orientation for cohort 3. An orientation program for cohort 3 is to provide detailed information on how you can apply and the benefits you can get. I believe the meeting so far, the presentation so far, have already made justice of some of the benefits and experiences you gain when you carry out, when you do internship with us virtually, right? Now, like I said,
Okay.
it's free, and based on the perspective you gave on research, that is actually the kind of perspective we follow in terms of our research internship program. Research internship program is not just about skill alone, but it's also about gaining research experience. And this project you just see, we're going to be publishing it. All right. So,
Okay.
and that is why every internship participants, we ask them to pay a commitment fee of $30 so that we can use those $30 to publish them, because we don't have any grants, we don't have any funding, and this is a biotech, one of the top biotech companies in Africa. All right. So definitely we need some funding for publication. That is why we're asking for commitment fee of $30. But you get to know more
Okay.
information about how you can actually join us and the link on how you can join as well. So definitely you get to know about it in my session that I'm just about to start right now. So I will answer a
Okay.
lot of your questions when we start right away.
Okay.
Yes,
Thank you very much. I appreciate.
you're welcome.
Thank you very much, our dear CEO, for enlightening us about things today and the questions regarding the questions he asked. So yes, we'll be having our wonderful CEO, the brain behind Genomac, to give us the orientation of new courts for court 3.0 now. Thank you so much.
Okay. Amazing. Thank you guys for welcoming me. I'll be sharing my screen right now. I'll be giving orientation to the new cohorts that we'll be having starting from next week. All right. So I'll be sharing my screen, and then we have detailed insight into what the program is going to be all about. So kindly indicate if you can see my screen. Uh, Moderator, please can you confirm if you can see my screen? Hello, moderator.
Yes, sir. Yes, we
Moderate.
can all see your screen. We can see your screen.
Okay, thank you so much. All right. Yeah, so this is orientation session for virtual research internship program at Genomac Corp. This is an opportunity for you to embark on a profound exploration of genomics and bioinformatics, and where you'll be able to master essential skill, foster global collaboration. The projects you just see being presented right now are actually powered by a kind of collaboration amongst participants from diverse countries. And you can see from the very beginning, a lot of people are joining from over 30 countries already. So it means that one of the key things about research internship is to expose participants to a kind of global culture, a kind of global teamwork, a kind of global team work experience, right? So because you never can tell what the future holds for you. So, and another thing we're going to be doing in the course of this research is show you how you can translate your research project into a publishable discovery of significance and impact. And the presentation so far is already uh, an evidence, right, of how our research internship is basically all about. So let's just dive in to have more understanding on the internship program. So here is what you get access to. So you get access to hands-on practicals. So you're not just learning, you get experience and exposure immediately. So it means this is a kind of a project-based learning. You are learning while carrying out research projects. So it's a kind of killing two birds with one stone. So it means like in the same course of the internship program, you are gaining essential skills to make you a researcher in your field of study, to expose you to research world, to expose you to in silico research world and genomics and bioinformatics. And you have a key understanding that genomics and bioinformatics is actually making a whole lot of exposure, making a whole lot of exposure, a whole lot of impact. It's actually making a waves. It's actually making a waves, right, on the biotech sector or in the biological related sectors, be it in the medical, in the agriculture, in the microbial sector, in the plant sector, veterinary, animals, humans. It's actually making a whole lot of waves. And it's causing a paradigm shift. So it means that just like data is actually being utilized in every industry, when it comes to every industrial sector of biotechnology, bioinformatics is actually gaining deep roots. So you also be gaining skills and expertise. You are able to develop your skills in bioinformatics, and everything you need from beginning to finishing your research project is being provided. You have a personalized mentorship. You can never get it wrong because you have a real expert to guide you. And you can see some of these projects that have been presented. Other key things is the fact that they have very good mentors. They have very good instructors, facilitators that guide them through the real life experience on real life data that we get from the public access data sample, and create the mentors help to create a kind of a strategic and a novel kind of research approach to the data, the general data set we get in order to be able to do a kind of a novel kind of discovery when it comes to and presentation and publication when it comes to carrying out this research. And then we have the tailored real life research project so that you are an expert after the program. So that I've seen people in the course of our programs where they did a general group with their group, like in teamwork, but still each individual of them were still able to apply whatever they learned to do their own research and also publish their paper. That's really amazing. And that is what you are able to do, and you can actually do in the course of this program at the end of the day. So it means we have publication and expert support to keep you in check. We have certification of completion and excellence that ranks you, right? That builds your CV and that places you out there for recommendation, that places you out there for opportunities like masters, PhD scholarship, and for you to be able to even venture in your career. Some of you could actually be a great, a stepping stone to open you to new job opportunity out there. And that's one of the key things we're actually looking at, because Genomac Institute Incorporated and Genomac Holdings at large is after impact. We are after impact. We are after making impact in the biotech sector, in the health sector, and across other industries as well. So we have a collaborative research paper ownership. So it means in the course of this internship program, you'll be a co-author. So your name will be imputed in the research paper while we're publishing, okay? And then we have recommendation letter for job opportunity, master's, PhD scholarship application. So there's needs around this. Needs around this must be communicated earlier. Okay, don't worry. We still have other perspective to how this research internship program is going to commence. So these are the current research projects we have for the incoming cohort 3A of our virtual research internship. So we have the human and other related research perspective. We have the clinical genomics and epidemiology research areas. Okay, so the first one, we have human cancer research. So we're going to be working on real-time human data. As well, Parkinson's disease, we are going to be working on real-time human data. So the colorectal cancer research and the prostate cancer research, we're going to be working with human microbial or microbiome genomic data, and then we're going to be seeing how these microorganisms are associated to increasing or causing progression in the metastasis of these cancers in human. And then we are going to be seeing how we can come up with identify specific biomarkers that can actually be utilized in the therapeutic approach in dealing or treating that particular cancer as well. So we have the clinical genomics and epidemiology. The first one is on the AMR perspective. AMR is becoming a global pandemic right now, and you can discover that see that a whole lot of pandemic or outbreak is actually happening out there, and some of these things has actually contributed to antimicrobial resistance in a very, very critical way. So how do we mitigate these issues? How do we solve these issues is what we are going to be looking at and working on the real-time real-time NGS data, either clinical, environmental, or across One Health perspective, and then show you how you can carry out research to mitigate these outbreaks and brings out about perspective to deal with that. Then we have the virulence genomics and regional pathogen surveillance. We have mobile genetic elements. Mobile genetic elements are actually one of the reasons why we have outbreaks and spread of virulence factors, of virulent mechanisms of antibiotics around, and causing quite a number of different treatment resistance to treatment. So we're going to be looking at that on the One Health approach, and we're going to be looking into cross-country comparative genomics of pathogens. Cross-country comparative genomics of pathogens. We want to understand what is the evolutionary dynamics and variations across different pathogens across different countries on a global scale. So those are really, really advanced research you can see in the course of this internship. So we have resources and access like lifetime access. So you're going to be given access to Genius software, but there's a yearly, the yearly cost of Genius software is $200, but we're going to be giving you lifetime access, and that is worth more than $5,000. So we have access to curated books and ebooks worth more than $5,000. Essential tools and resources will be given to you as well. And then customized research project will also be given to you. Now, one of the key things I want to announce to all our research internship, even the previous ones, all right, even the previous research interns, is that we are happy to announce to you our automated research platform called Omics Board. Omics Board is a one platform for exploring the world of omics sciences, where you discover curated data set, video tutorials, hands-on assignment to turn project into publishable research. So you can discover some of the challenges we face with internet right now in the course of this program. So everything that needs to be taking place in course of carrying out a research from basic to advanced, carrying out projects, gaining hands-on experience, skill set, and also getting insight, translating all your findings into a publishable research paper is on Omics Board. And while we have Omics Board, we also have real-time experts in place to be meeting with you once every week for practical guidance, for expert mentorship, to guide you, to give you perspective into carrying out the analysis and translating into a research paper, the publication aspect and all of that. So it means we have two things, two powerful pillars to make sure that our research program is successful. You may be having doubt that how can people all over from different countries be able to carry out research on genomics and bioinformatics. Internet has made it possible, but we have challenges in internet in the course of probably network not being available. So or maybe we get lost of videos and all of that. That is why we come up with Omics Board. So on Omics Board, you have access to, please, are you all with me? Let me try to confirm. Please, are you all with me? Moderator? Amazing. Thank you so much. So that is why we come up. We have built Omics Board, an automated research platform. So on it, you get access to data set. On it, you get access to video tutorials from your mentor, all right, to give you practical illustrations on what you need to do actually on a step-by-step approach from A to Z. From carrying out research analysis to collecting the research analysis to doing the statistical analysis to come up with visualization, down to research thesis writing on in terms of the introduction, materials and method, abstract, putting materials and method in place, results, discussion and conclusion. Every step modules have rights up. You understand my point? So if you are doing analysis for data collection, you're in the same platform. You also have what's it called right now? You also have a place where you learn how to do the materials and method, how to come up with rights up for data collection under the material method. Now, same thing, you learn how to put maybe visualization or tables together for results. You also learn how you can translate that result into a publishable insight. Yeah, into an insight that can be publishable, and also you learn how to report it in a results way. And you're going to be doing this with a group with a group of people. So it's a kind of teamwork. All right, it's a kind of teamwork. And I just want you guys to have understanding that this project that we're just presented, we're going to be publishing them in the nearest possible time after all the work has been collected and put together. And that is one of the reasons why in the course of the internship program, we usually request for a commitment fee, because we don't have funding. Yeah, we don't have funding. We are not collecting money for the experiences, for the skill set, for assessing Omics board. And now we are partnering with Omics board. Are you getting it? Omics board, to get access to research platform, Omics board costs $1,000. And we are covering all of these things. All right, we're covering the software tools. You get access to the software tools. Are you getting it? You get access to everything you need. All you just need to do is pay a commitment fee of $30 in order for us to be able to put some resources together and also compensate our instructors, and as well put resources together for publication of this 8th project by the time we're done. Are you getting the point now? So, and we understand equity. We understand people being able to get access to the same information. And that is why we're doing all our possible best as an impact-driven company to impact life and raise giant researchers out there who will make tomorrow better. We are contributing to the next generations, and that is why we're doing this. All right? So please try to see the impact factor of what we are doing, and the novel research aspect of what we are doing, and the whole of the benefit you are going to be getting down to recommendation letter, certification, the experiences, the skill set, the software tools you are going to get access to, the experiences you are going to get with expert mentors in the field, and all of that. So it's quite a lot. It's priceless. It's invaluable. Are you getting my point now? So you cannot miss this by the time you are selected, because we usually selected the participants. We already have over 500 or maybe close to 600 applications already. All right, for the first cohort, and we're going to be selecting people from this cohort. And already we have applications from over 40 countries globally, all right, both in America, Asia, Europe, Africa. So what am I trying to say in essence? What's it called right now? How Omics board operates. All right, I want to show you how Omics board operates. So I'm taking you to Omics board. I don't know. I hope you can still see my screen. Hello, can you still see my screen?
Yes, we can, sir.
Okay, so this is how Omics board looks like. And as you can see, this is project classes. There are other fundamental classes that you're going to be gaining. And you're going to be taking through the basics of genomics and bioinformatics, where you get access to software tools and perform basic fundamentals, genetic sequence analysis to prepare you for the project classes, right? So, but apart from that, this is the project classes. And you can see from module 1 to the rest of the modules, A to Z of the project work. Like I said, you're going to get access to mentorship once every week with an expert mentor to guide you as well. But for you to carry out research project in a group, everybody has a task to do. And for each of the tasks, you are shown what data to carry it out. Are you getting it? And once you carry out those analyses, you are also given tasks on materials and method. All these things will be taught. We'll show you how you can navigate your way with Omics board, the tasks that will be given to you, the videos you need to watch to learn how to do that task. While you're watching those videos, you are getting skills. Because the past cohort, we had a lot of challenges with network, with meetings, instructors at times not being available because of one thing or the other. But everything you need to do, all the tasks you need to do, how you need to do it, is already on Omics board. Like I said, Omics board costs how much? $1,000 in the pricing plan, but you are not paying a dime. You are only paying out of access to Omics board. You are not paying. You are only paying the $30 for the publication resources and putting some resources together also for instructors. Are you getting the point now? So we have the result aspect where you have where you are going to upload your work so that your group leader will be able to collect and works together for the research. I mean, for research publication purpose. Like I said, everybody get tasked what to do, how to do it. I want you to perform the analysis, how to translate it to materials and method write-up, how to translate it to result write-up, and the discussion and conclusion write-up and all of that is already on Omics board. Are you getting the point now? And we're going to be giving you guys access to it. Why you have a weekly live access, like live class access with mentors to guide you to attend to your challenges, to attend to issues and put some issues together, to give you access to data samples, NGS live access, real life data sets to carry out your research project and all of that. Isn't this amazing, guys? What do you think? And I need a reply at the chat room. Can you guys respond quickly? Yes. Yes, it's indeed impressive. It's indeed impressive. Yeah. Thank you guys. So that's it for the orientation. And let me just try to chip in a bit. Like I said, all the selected participants that we're going to be selecting on the course of this new cohort 3.0A, you are going to be paying a commitment fee of $30. I already tell you what that $30 is all about. Okay, I think I've been able to explain myself clearly. So let me just give you update on the date. So between today and tomorrow and Sunday, we are going to be getting, all selected participants are going to be getting their selection on email. So those of you who maybe you are interested in the research internship program, let me show you how you can apply. So go to our website, genomacop.com/online-.com, right? .com/online-internship, okay? /online-internship. Okay, so this is the platform, genomacop.com/online-internship. And this is the is where you are going to register. You get access to every available information you need to get. So we already came up with a research topic, research aim and objective. And this is where you can download the brochure to see the full detailed, comprehensive information on each of the research programs to carry out. Now, I would like to say that the previous IT interns, and the course of the previous course, you guys really did amazing. And I want you guys to have an understanding that we'll be publishing your work very soon in high impact research journals. And we're going to let you guys know on your respective research group, okay, on the rest of the details, okay? And like I said, we had a challenge of a lot of challenge in the course of last time internship program, but right now we came up with Omics board. I want to show you what Omics board is all about, and that sort of a challenge will not work. In fact, this research internship is going to be running for 9 weeks, and the 10th week, I think it's on the 11th week or so, or the 12th week, we're going to be having graduation like this, where you present the core 3, we present their project, and they will get their certification, and they're going to wait for the publication of the work as well. So that's it. Do we have any questions so far? Do we have any questions so far? Do we have any questions so far? Do we have any questions?
I think
You can unmute your mic to ask your question, please, one at a time. Anybody with questions?
so. I think you have answered my question, my previous question. Your explanation have answered everything to me. And of course, this is a great achievement for the younger researchers. In fact, and even not just younger ones, but even the PhD students. This will be helpful to them. Thank you.
Exactly. Exactly.
Yeah,
You're
because
welcome. Thank you so much for the comment.
world is moving toward bioinformatics. So most of the data are available and they were there at our disposal. So we can use computational data tool to like have a clinical data, biological data, like in form of 3D, three-dimensional structure. And of course, we can study it like all this in silico method. Thank you very much. We will look into it.
Thank you so much.
Exactly. Amazing. Thank you so much for the comment.
Thank you.
Thank you so much for the comment.
Thank you very much, dear CEO, for this beautiful orientation. I'm sure we can all say what it is all about. Please, you have questions. Please start.
Yeah, hello, good evening.
Good evening.
Sorry, my name is Nana Safo from Ghana. Yeah, thank you so much for this opportunity. It's a great stuff that you have given us this evening. My first question goes to Mr. Oliwasi. So you are now introducing OMIS board in this intensive program for the first time, right? Yeah, hello, can you hear me?
No, no, no,
I
no.
said,
We have, yes, I'm with you. We have different programs that are going on Omics board. All right. We have research scholarship program. We have research fellowship program. We have come up with a research program for internship, different from research fellowship, that is specific to the kind of project they are going to be carrying out. Are you getting it? So Omics board is a kind of complex, a very broad place. Are you getting the point now? Hello, are you with me?
yes, I'm with you. I'm listening.
Yes, yes. So Omics board is a broad, is a platform with a broad area of research. Are you getting the point now? So the research internship, we already came up with a curriculum for them to be able to carry out their research program on Omics board, specifically for internship, different from every other programs that we are running. I hope I'm
Okay,
able to
so
answer your question.
all right, so those who just presented, they did not go through OMIS board, right? But the 3.8 people.
No, no, no. Yes, yes. Yes, because we didn't have Omics board when the previous interns were doing. And one of the reasons why we came up with Omics board was the fact that there were a lot of challenges while we're doing the online classes. Some were not available, at times we lose access to video recording. That's why we came up with Omics board, also for internship, to be able to get access to what is specific for them, specific for research internship based on the programs that we have structured and the curriculums.
All right, all right. That's good. That's brilliant. Yes, my next question is how can one those of us who are still doing the fellowship program, how will that be different from this internship program? And won't one mislead the other? Is it not a prerequisite that you finish the fellowship program before you do the intensive program, or is it
Of course.
can
No. The thing is, if you have the capacity to be able to do that, all right, if you have the capacity to be able to do that, research internship classes is always on Monday, right? The lifetime access is always on Monday. So if you have capacity to combine both, why not? Are you with me?
Oh, okay.
Why not? So because the classes is not per week, it's not the same day. All right, it's not the same day. Research internship classes is always once a week, and that is on Monday. All right, so that's the basic. Thank you.
Oh, okay. Okay, so the internship is not a prerequisite. So the fellowship is not prerequisite for internship, right? You can do the two at the same time.
Yeah, if you have the capacity to do that, why not?
Oh, okay. All right.
So thank you. Moderator, please, can we go to the next section because of our time very quickly so that we can
Please,
have
I have a
other
question.
project presenters? Oh my, because
Please.
we need to. Okay, go ahead.
Okay, please, I want to find out. This is my first time of joining this platform. Where can I start from as a
Okay.
beginner? Yes, as a beginner. Or can I join the internship? Or where can I start from as a beginner?
Yeah,
Or
yes, you can join the.
though I have some experience in this thing, can
I
I join
understand. I
internship
understand.
direct?
Of course, of course. I understand the perspective of why you say internship. There's some internship that you need to have skills for before you enter. But here, you go through the basics to the advanced, so you don't have any worries. Okay,
Hello, sir, Mr. Shei.
I'm with you.
All right, thank you so much. I just want to make a little comment and perhaps ask one question. Thank God
Okay,
for what you are doing. I was a beneficiary of your platform sometimes early last year, and I learned
okay.
phylogenetics courtesy your platform. I want to give thanks to God. You know, when you came on board, we were like, that's something is actually wonderful new, but I've seen that you have really much advanced to tell us that there are plenty other things on board for us to enjoy. I want to salute that innovation. All right,
Thank you
my
so much.
question actually is that considering the diverse research areas that you have presented to us, all the skills to be learned in this period of internship, does it cut across these diverse, what's it called now, research areas? Take for instance, okay, I'm to learn metabolomics, I'm to learn these transcriptomics and all of those other tools, bioinformatics tools. If I subscribe for, take for instance, antimicrobial resistance, do I still have the chances of learning all those listed skills? That's just my question.
In the course of 9 weeks. No. Now, remember, we say this is research internship. It's specifically research. So when you pick an area, we want to make sure that you are very, very good in that area. Now, let me tell you something. By the grace of God, I'm the founder and CEO of Genomac Holdings and its respective subsidiaries. All right. So, but you see, I've been in this field for the past 7 years. Are you getting it? And everything I know so far in the course of genomics and bioinformatics took me 7 years to do. Though the reason why it was like that, like when I say that I'm very good, so I'm very good in different areas. But when I started, I didn't start by learning everything. I just know everything like that. Are you getting it? So one key thing with genomics is that start from somewhere, and each thing you are learning, know it very well. It will take time for you to learn diverse areas of bioinformatics, but each area you are gaining, know them very well. And that is why we want people to really know it, so that within a short possible time, you'll be able to grasp all these things. So are you getting my point? So when you pick antimicrobial resistance, we'll show you how you can be very, very good from not just the analysis alone, but even to the research publication aspect, even to report aspect, being involved in the research experiences and all of that. So that's it basically. Yeah.
All right. Thank you so much. I can attest to that.
Hello, sir.
Based on the previous experience, actually.
Hello, sir. Sir, I have a question.
Yes, thank you.
Can you tell us, please, about the benefit?
Sorry, we can't hear you. I think your network.
Yeah,
Is it your network?
I repeat
I
again.
think it's your network. Hello?
Hello, sir. I have a
Go
question.
ahead. Go ahead, please.
Yeah, my question is that
yes.
as I am a student of BSc, and what will be the benefit for me of this internship?
Okay, please. All the benefits is on the research program. Is on the WhatsApp group. So please, can you go to? I will send the link right now. Okay, I will send the link to the website. I will send the link of the website to the chat room. Can you go through the link? All the benefits are there. All right. All the benefits you are going to get as an undergraduate is there. So just go to the chat room. I'm sending the link to the chat room now. Kindly use it. Yeah, thank you. So please, can we go to the next section? We don't have much time. If you have a question, just drop it at the chat room. We will answer your questions before the end of the meeting. So just drop it at the chat room. Thank you. So go ahead, please.
Thank you very much, our very own CEO of Genomac. Thank you for bringing up Genomac into existence. Thank you very much for this great opportunity. I, myself, and many others are very glad to be part of this. So going on, we would invite one of our very own, Miss Moyosore, to give us the testimonials on past participants that have joined our internship and what they have to say. Thank you very much. Miss Moyosore, can we have you on board?
Good day, everyone. I'm Camille. Thank you all for joining, and congratulations once again to all graduates from the internship program. I'm trying to share my screen. Can you hear me?
Yes,
Yes, we
we
can
all
hear you.
can. You
Yes,
can
go
go ahead.
ahead.
Yes, we can.
All right. So thanks for staying for the section thus far. So
F, please kindly mute your mic if your mic is on. Thank you. Abdul, can you please? Thank you very much. Please kindly mute your mic. You can go on.
So I'll be sharing some testimonial from some Genomac students that graduated from the internship program. First on the list, I have Mr. Akash Balajit Singh. He graduated from the Institute of Science and Technology in India with a BTech. He said, I worked on genomics of food, biome, infectious diseases. It was really amazing working with you. I really loved it and gained more knowledge by connecting with people across the globe. The skills that I learned here are really appreciable. In real life, if I put this on research, I can work in several other fields to bring advancements by applying my own practical knowledge that I have gained by this internship program. Working with everyone was quite fun and I learned a lot of new things. That's from Mr. Akash. We have Mr. Farouk Adeyemi Yusuf. He graduated from the University of Lagos with a BSc in microbiology. He said, I worked on functional insights into microbial genes associated with pancreatic cancer promotion and progression, a metagenomics approach. My journey with Genomac is truly one of those moments in life where you get to do the right things at the right time. I had high hopes and aspirations regarding what I was going to get from the internship, and I was not disappointed in the end. I was inspired to join this program because I saw a repost of one of their programs from a respected mentor and supervisor of mine who happens to be a professor. The knowledge I've gained from this internship will allow me to tackle specific wicked problems that are of interest to me, like health and environmental problems. That's from Mr. Farouk at the end. I also have from Mr. Gima Edward, a graduate from the University of Science and Technology in Ghana. He graduated from a BSc in biochemistry. His testimonial goes thus. I worked on identifying single nucleotide polymorphism in breast cancer patients. This educational journey has been a pace setter for me. I was keenly interested in bioinformatics, but had no chance to learn the right resources until I joined Genomac. Genomac, through this internship, has helped me to be able to navigate on biological database like NCBI GenBank. After this internship program, I have been able to do analysis of biological data, and moreover, I have been able to navigate my way out into achieving my goal. I love bioinformatics. That's from Mr. Gima. We have another one from Ms. Akinyemi Mary Anoluako, a graduate from the University of Osun State in Nigeria. She graduated with a BSc in microbiology. I worked in computational exploration of phytochemicals from traditional medicinal plants for predicting anti-malaria activities. I have always wanted to work with like-minded people on research, and this was exactly what I experienced. Now I can say I know what a team spirit is. The knowledge I have gained will help me further research in discovering new drugs, which will help me in fighting the issue of drug resistance. We have another one from D. Siri Sivani. She's a graduate of the Institute of Science and Technology from India, a BTech in biotechnology. She said, I worked on genomics of foodborne infectious diseases. I personally felt amazing when I was working with this team. I felt like I was developing my skills by reaching people and learning something new. Every time I learn new things, as people across the globe have good ideas stuck in their heads, it was quite interesting till the end. The skills that I learned here are well appreciable as everything is new for me. As we know, gaining knowledge can give enough ideas. Using this knowledge, I can contribute to my own field by deploying new ways to work. And from the last person, we have Mr. Yusuf Abdulatif. He graduated from the University of Aigba, Kogi State in Nigeria. He has an MSc in anatomy. Prince Abubakar, that's his name. His testimonial goes thus: I worked on Staphylococcus aureus. I have always been eager and longing to acquire skills in bioinformatics. The idea of in silico research using laptops and online tools without going to a physical laboratory fascinates me. When I heard about the Genomac internship program, I grabbed the opportunity and I'm glad I did. During the internship, I was introduced to several bioinformatics tools, how to search and navigate bioinformatics sites and analytical tools I wasn't aware of before now. So that's the testimonials we have from some of Genomac students that have gained in one way or the other from the internship program. And we can all see that the programs are not, they're not restricted to one thing, and there's a lot to learn from the cancer research to the bioinformatics. There's a whole lot to learn. Finally, nothing in life is to be feared. It is only to be understood. Now is a time to understand more so that we may fear less. By Marie Curie, Nobel laureate. Thank you.
Thank you very much, Ms. Muyasura, for these testimonials that we're getting from the past students. And if you've been with us right from the beginning of this present moment, we want to say thank you for staying with us thus far. I'm sure you can see the great things that Genomac is doing, from the various presentations to the orientation course by our own CEO to this testimony. I mean, this is awesome. This is awesome. That's all I can say. So just to round this up quickly, we'll be having a live section of testimony from one of our past students, Mr. Farouk. Mr. Farouk, please, are you on this call? Can we have you for your live testimony section? Thank you.
All right, so can we have, please, can we have number 4? That would be Sweba. Sweba, are you here? Sweba, are you here? Okay, can we have, I think Sweba is about to join. Sweba? Hello, Sweba, are you here now? Okay, can we have number 5? Number 5 is Mr. Bartholomew on drug design and discovery. Hello, Mr. Bartholomew.
Can we have Mr.
Hello,
Bartholomew
yeah,
on
good
board,
evening.
please? Thank you. Please, you have 5
All
minutes for
right,
this
I'm trying.
presentation. Thank you very much.
You're welcome. I'm trying to share my screen. Okay, can you see my screen? Hello.
Yes, we can see your screen. Please, you can go ahead. You have 5
Okay.
minutes. Thank you.
Okay. Yeah. Good evening, everyone. My name is Bartholomew Kolo, and I'll be presenting on behalf of my group. So worked on my group is drug design and discovery. So our research topic is computational exploration of phytochemicals from traditional medicinal plants for predicting anti-malaria activity. So as we all know, malaria is it's a major global health issue, particularly affect the tropical and subtropical regions. And we also know that despite the efforts to control it, about 229 million cases and 409,000 deaths have been reported worldwide in 2019, and it's increasing day by day. So the recent computational methods have revolutionized drug discovery by enabling rapid and cost-effective screening of bioactive compounds. So this paper combines computational screening with experimental validation to discover new anti-malaria agents using molecular docking and discovery studio software. And then this study aims to identify promising candidates from traditional medicinal plants, accelerating the development of effective malaria treatments. So the biological activities reported for this study will be B. saligna, which include antiplasmodal, acetylcholinase, and then alpha-glucosidase. But the compound we are going to focus in the course of this study is going to be alpha-glucosidase. So what's the research aim? The research aim is to computationally explore phytochemicals derived from traditional medicinal plants with the goal of predicting the anti-malaria activity. By employing advanced computational techniques, this study seeks to identify potential natural compounds that could serve as an effective malaria agent, contributing to the development of novel treatments in malaria. So our research objectives are optimizing malaria drug discovery using computational techniques and validating predictive models for anti-malaria compounds. So what are the materials used for this study? We use the phytochemicals database, a curated collection of compounds. A curated collection of compounds from traditional medicinal plants with reported anti-malaria properties. So the alpha-glucosidase, that's a compound that goes from Budenglia saligna, is actually we underwent some literature review on it and then found out that this alpha-glucosidase has anti-malaria effects. So we have different software that we use for it. So we use the Discovery Studio, which is used for molecular docking and analysis of ligand-protein interactions. We use the Autodoc. We use it to predict how small molecules and drug candidates bind to a receptor. Then we also use the PDB, the Protein Data Bank, which is an open access database that saves and distributes 3D biomolecular structure information to assist in the exploration and design for therapeutic and diagnostic equipment. Then we also use the PyMol, we use the PubChem to discover chemical probes through high throughput screening of small molecules. We also use the protein targets, which is the key malaria-related protein targets for docking studies. So what are the methods? The first thing we did was to select our phytochemicals. Can you hear me? I want to be sure I'm not speaking to myself.
Yes, we can
You can
hear
go on.
you.
Yes,
Please, you have
you can go
15
on.
minutes
Okay, all right.
left. Thank you very much.
All right, thank you very much. Okay, so the method, we have the phytochemicals. So just like I said, we have to visit the literatures in order to get our phytochemical, in order to get the phytochemicals, yes, that we use. And then it's not just about getting the phytochemicals, but getting the active, the active compound that works in the course of getting the phytochemical. So we have the pharmacophore modeling. And then also did the molecular docking, which is like the most important thing, where we imported the phytochemicals and protein targets into Discovery Studio. We performed our docking studies to predict the binding affinities and interaction modes of the phytochemicals with malaria protein targets. And then we also did our in silico screening, which involves performing an in silico screening of the matched phytochemicals that we used against the pharmacophore model to identify those with the highest potential anti-malaria activity. So also did a virtual screening where we have to screen the phytochemicals for their potential anti-malaria activity and identify promising compounds for experimental testing. Okay, so after our docking, so we had our clean protein. We had to clean our protein before docking, by the way. Had it been I'm presenting this live, I could have kind of rotated these proteins for you. But then when you see the protein, you can see the amino acids located at different ends of the protein. You can see the leucine, the valine, and different amino acids that attached to this particular protein. So these are our results. this is our protein, and then if this is our protein, and if you can look deep, you can see our ligand inside. Yeah, just like I said, had it been in my software, I could have rotated this protein so that you see how the ligand binded to the protein. So yeah, in conclusion, in this study we utilized autodock to perform molecular docking of alpha glucosidase. Alpha glucosidase derived from Bulegia saligna plants against key malaria protein targets. So our docking protocol was validated through redocking experiments. We had to redock and make sure that all got was successful, and then we successfully reproduced knowing binding modes of established inhibitors. Then the binding affinities of the docked phytochemicals indicated promising interactions with several compounds showing low binding free energies, suggesting stable complexes. So which this means that alpha glucosidase is actually a very good ligand and can be further studied to see if it can be able to elucidate some efficacy in the treatment of anti-malaria. So now we want to express our heartfelt gratitude for the incredible internship
Mr.
experience we had at
Bartholomew, thank
Genomac.
you very much. The time is how is no longer on our side. Or can you please round up in just 1 minute? Thank you.
Yeah, sure. So, yeah, we experience our heartfelt gratitude for the incredible internship experience we have at Genomac. This opportunity has been invaluable in shaping our professional journey and has provided us with skills and insights that will undoubtedly benefit our future career. We also thank our facilitator who has always been there for us, meek and humble, and make sure that we all participated in the internship. So that's the end of my presentation. Thank you.
Okay, thank you very much, Mr. Bartholomew, for this beautiful and wonderful presentation. We're so glad that you're part of us, you're a product of our great research and innovation. Thank you so much. So we'll be having candidate 6 on board. Please, if you're on, kindly signify. You have just 4 minutes for your presentation because we don't have time on our side again. Candidate 6, can we have you on board?
Yes, I am online. Hello.
Yeah, you can go ahead.
Thank you.
Please, you have strictly 4 minutes for your presentation. Strictly 4 minutes.
Please, can you see my slide now?
No, your slide is not visible, sir.
Okay. Sorry. Can you see it now?
No.
Hello, can you see it now?
Visible, sir.
No,
Still not visible. We still can't see your slide.
Can't see anything.
Okay. I'm sorry. I was trying to share the screen, but I don't know what's happening. Yes. Can you see it now? Can you see it now?
It's still not
No,
visible.
no.
Yes, it says we can see it now. Please, can you use the full screen of yes, we can.
Can you
This
see
is
it
good.
now?
Thank you.
Okay.
We
You
can
cannot
see it.
see
You
it.
can go ahead.
I cannot
All
see
right.
anything here.
Mr. Abdul, can you please probably log out then log in back? I think the screen is clear for everybody. I mean, can you see the screen? Please reply in the chat room before we go ahead. Please let us know in the chat room. You can see. Okay, thank you very much.
Yes.
You can see your screen. You can go ahead, Mr. Azaz. Please, 4 minutes for this presentation. Please do not exceed
Okay.
4 minutes. We do not have time on our side.
Thank you.
Thank you.
My name is Aziz Dada Shola, and I'm to present on Unlocking Nature's Secrets, the incredible evolutionary study. Okay, thank you. Introduction. I collected different sequences of Wabachia in mosquitoes and the black fly, being the major factors of serious diseases. Malaria is inclusive, dengue fever and the likes are also inclusive. And This Wabachia pipens majorly has been reported in different paper that they actually reduce the vectorial capacity of some insect vectors. And that's the reason why the major aim and objective of this study is to understand the knowledge and the role of that Wabachia in mosquitoes and black flies of different regions. And to also study the phylogenetic association between Wabachia and the two factors, I mean the mosquitoes and the black flies. And my justification, since no report has been made on the study of phylogenetic relationship between Wabachia of mosquitoes and black flies, I think there is need to also explore and have an in-depth understanding of what the role of Wabachia is in the two factors, I mean mosquitoes and the black flies. However, my materials and method include we collected 41 nucleotide sequences of Anopheles mosquito and Salmonella species. The Anopheles mosquitoes, especially Anopheles gambiae, is the major vector of malaria parasite, while Salmonella species, I mean Salmonella damnosum majorly, is also the major factor for river blindness, that is onchocerciasis. And these nucleotide sequences that we collected were of less than 1,500 sequence. Then we did see the sequence alignment making use of Geno software, and mapped aligner tool was used simply because it has the opportunity to identify the direction of the sequence and do the alignment proper. And these are the information of the sequences being collected. Some were from Ghana, while some were from Belgium, Myanmar, India, and the likes. You can see it in this picture. Now, and we use the sequence aligned to construct the phylogenetic tree, and the result is as follows. This is the alignment view, where we were able to see some different consensus identity view of the sequences. You can see the first almost 12 or 13 here, they have almost similar sequences, while there are some deletions here, and we also have it in other way. And this is the phylogenetic tree showing what we observed. Then to the first rate that we have here, if you can see my cursor here, the sequence MW179597 is a Wabachia sequence from Anopheles gambiae. And this one, the one that ends with C02 on the red column also, is also Anopheles gambiae. And these two were sharing almost 84% similarity with our Wabachia sequence from black fly, that is a Simolium squamosum. However, if we start with the clade that we have here, you can see we have almost all Anopheles gambiae are the first 12 that we have here. So, but Anopheles gambiae find its way down to the Wabachia sequence of Simolium squamosum, which is from Ghana. And the first two red here were from Guinea, while the sequence that ends with 995 is also from Belgium. So you can imagine having Wabachia sequence from a particular mosquito having its way, I mean its similarity, some similar sequences with that of the squamosum in Ghana. So, and if we try to look at this map, I think it's showing the distance between the different regions across the study area. You can see Belgium is here, very, very far apart to other countries here. And like I said earlier, we have some sequence from Belgium that is very close to that of the Simonium squamosum in Ghana. And Ghana is here. You can see the legend, it interprets the map better. So, and this is the heat map of the sequence, where we have just the different similarities of those sequences being displayed. Now, discussion. Sequence MT449 from Thailand and sequence 805 and sequence 806 were from Myanmar. Although Thailand and Myanmar, they are including India, they look very much more closer. And this may be as a result of the closeness of these countries to each other.
Mr. Aziz, please, you have 1 minute more. Please kindly round up your presentation. Thank you.
Thank you. So in conclusion, similarities between Wabacha sequence of countries in the same continent may be expected due to their closeness to each other, while that of the countries far apart may be eccentric and unexpected, but nevertheless still occur. Likewise, the similarities and evolutionary relationship that exist in sequences of anomalous species and familiar is also outlandish. Therefore, this study suggests further research on how Wabacha can be used for vector control of malaria and onchocerciasis, as it is known to reduce the vectoral capacity for disease transmission. Thank you.
Thank you very much, Mr. Aziz, for this beautiful presentation. Please, can we have candidate 7 on board, presenter 7? Please, are you for? And if you're on here, kindly signify. Presenter 7. The 7th presenter, are you on board?
Yes, I'm on board.
Okay, please go ahead. You have strictly 4 minutes for your presentation. 4 minutes.
Okay, I'm trying to share my screen.
Mr. Aziz, please can you mute your mic?
Can you see the screen?
Yes, we can see your screen. Please can you use the full screen option?
Okay.
Mr. Aziz, please can you mute your mic? Thank you.
So this is the Cancer Genomics Group. Our topic was based on investigating the functional impact of SMP-associated variations in breast cancer. And these were the aims. We aim to comprehensively investigate the functional impact of single nucleotide polymorphisms that are associated with breast cancer. And by analyzing this data, we are able to understand how single nucleotide polymorphism plays a role in the development and the progression of breast cancer. So the objectives was to invariant characterization to identify, to annotate and analyze the frequency and potential functional effect of SMP variations associated with breast cancer, and to establish some clinical correlation between SMP variants and clinical outcomes. So as we all know, breast cancer is the most malignant tumor in women worldwide, and as advancement in genomic research continues, the role of SMPs has been much more important now, and as a relatively small allele, SMPs has a high availability and linkage disequilibrium, which exists among different populations. So that shows the importance of SMPs in the prognosis detection of breast cancer. So the materials and method we got the data from NCBI, and then both the reference and the query data was gotten from NCBI, after which we moved towards quality control, in which we assess the quality of the data, and then we filter out any low quality reads, and after which we assembled genome, which was later mapped to the reference. So when we did this, after this was done using
Mr.
a
Timtokwe, you have less than 2 minutes more. Thank you.
this was done through the Genius software, and our analysis revealed that there were 191 instances of deletions, and out of that 191, we had 116 that were tandem repeat deletions, and insertions were over 2,000, and 881 of them were tandem repeat insertions. Insertions are able to, alter the gene structure and impact the function of protein and even gene expressions. And more importantly, the single nucleotide polymorphisms we discovered were transitions SNPs, and there were 89,641 transition SNPs that we discovered. And then for transversion SNPs involving purine to pyrimidine, or vice versa substitutions, there were over 41,000. For substitutions, we had 4,258 occurrences, out of which 40 were associated with amino acid changes. So this is just a little graph showing in
Please
percent.
kindly round up your presentation, Mr.
So,
Timtokwe.
okay, so SNPs are the most common genetic variations related to breast cancer, and it is important that these SNPs are, studied further to, understand the individual dispositions to HMP, to breast cancer, and then not just that, so that we will be able to integrate our results with clinical outcomes, and ultimately to help us with breast cancer prevention and detection and treatment. So thank you for the opportunity to be part of Genomac. It's really an opportunity. Thank you so much for the skills, for the knowledge, and the hands-on experience. Thank you.
Thank you so much, Mr. Timitope, for this beautiful presentation. It has shown things that you've done in Genomac and how Genomac has affected your research lifestyle. We're so glad to be a part of this. So we'll be having candidates 8 on board. Candidate 8, are you on? The presenter 8.
Yes, ma'am.
Number 8. Okay. Please, you have exactly 4 minutes for your presentation, please. Thank you. So we can round up on time. Thank you so much.
Hi, myself Hasana Ibrahim. I'm here with the topic mapping cross-species transmission pattern of foodborne infection. Foodborne diseases are the intoxication and infection caused by the pathogens like Salmonella, Shigella, Campylobacter, E. coli, etc. And this causes 1/10 of the mortality, and this given by reported in CDS in 2012. And by Brian in 1982 reported that more than 200 infectious diseases can be transmitted through food. The problem is made worse by the continuous threat from emerging pathogens that can evolve to adapt to the different environments resulting from ongoing changes in farming or production. Genomics provides new insight into mechanism of bacterial evolution and helps in determining virulence factor of pathogen. The main aims and objective of this study is to explore and analyze the pathogenic traits present in various strains of different Campylobacter species, and also cross-genus comparison and evolutionary relationship with these different species. Also analysis the antimicrobial resistance and virulence profiling of the selected strains. Materials and method. we collect the data from the NCBI. We collected almost 25 assembled genome of Campylobacter jejuni, Campylobacter coli, Campylobacter upsaliensis, and Campylobacter fetus. We done and genome annotation with the RAS tool kit on Patrick annotation pipeline. Then predicted open reading frame functional categories in the genome was using the NMPDR. It is a seed-based prokaryotic genome annotation service. Then done the comprehensive genome analysis where the chromosomal property is compared with the genome strains, and we end up with a genome map, a circular view using the bricks. Then average nucleotide identity analysis and phylogenetic analysis with the help of J-Species WS pairwise genome comparison web server. And phylogenetic tree was done with the Patrick tree builder. Then analyze the resistance and virulent factors of this species using BLAST-P and KMR based detection method for microbial resistance gene. Here are
Miss
the results of
Farsana, you have just 1 minute left, so please kindly conclude your presentation. Thank you very much. And presenter Nai, please start getting ready. Thank you.
Okay, ma'am. Here the result of Campylobacter coli. You can see that the red in color, which is the highest seen strain, that is 142401 AMR gene and highly expressed in the JZ1. And the figure 2, it is sample against antibiotic class. Here the red in color shows the sorry. It is a figure of sample against antibiotic class. All strains, this all strain possess the resistance gene to all antibiotic class with the expectation to peptide antibiotics, cephalosporin, isocyanide, lincosamide, streptogramins, phenicol antibiotic, rifamycin, oxazolidine, which only in 18Q. 18QD 2YX 296. And this show the sample against antibiotic. In here, strain 1424.0.1 has shown the high resistance. Also in the figure 4, sample against resistant mechanism. In here, all strains prominently employ antibiotic targeting suspectable species and the efflux pump resistant mechanism. The figure 5 shows the
Okay,
gene
I'm
against
sorry, Miss Farsana, please, your time is up. Please, can you just go to the conclusion? Please. Thank you.
Session and conclusion. Strain 142401 1697 99 18 and JZ1 shows the highest level of resistance. Strain 80QD 2YX296 is resistant to the highest number of antibiotic class, which shows infections caused by this strain will be difficult to treat. All strains predominantly employ antibiotic target in susceptible species and efflux pump as a resistance mechanism. Majority of the virulence gene encode for protein that function in motility, chemotaxis, invasion, and phase variation. Strain Canton B4 NCTC 11366 have the highest number of mechanism to exert their pathogenicity, and also strain Canton 69791820190916H and LMG 9860 strain have the most abundant virulence gene, many of which may be involved in a single virulence mechanism. And the conclusion here is that antibiotics targeting and antibiotic targeting is in susceptible species and efflux pump may be a good antibiotics to treat infections caused by Campylobacter coli. peptide antibiotic cephalosporin isocyanide, lingosamide, spectrogramin, phenicol, antibiotic rifamycin, oxazolidine may be the most suitable to treat foodborne disease caused by Campylobacter coli. Here are the references.
Thank you so much, Ms. Rastana, for this beautiful presentation. Please, is candidate Nai, presenter Nai on board? Can we have presenter Nai? Are you on? Presenter Nai, are you on? The last presenter for the day. Okay. If we can find her, we'll be moving on to the next agenda. Once again, a very big article and congratulations to our graduates that just concluded course 2.0, virtual internship. We're glad to be a part of your research journey of your innovation into bioinformatics, and we're really glad to be a part of your research journey because without you, there is no Genomac. Thank you so much for being a part of this beautiful journey and path. Your certificates will be dropped to you via WhatsApp. Every graduate is going to be awarded certificates to show for the training that they went through while they were with us. So your certificates are going to be dropped to you via WhatsApp. Then I mean you can also agree with me that this has been a very wonderful program, a very exciting, innovating program from the opening remarks from our director of institutes to our CEO's address to our CEO's address to the presentations that our fellow graduates have had, our past participants have had on the researches that they've carried out. I mean, it's awesome, it's awesome, it's awesome. We can say that they've been doing a very great job ever since they've joined us. And I'm sure for the new cohorts 3.0, I want to encourage you all to take your lessons right, take a pen and paper, jot down everything that you will be taught because you're going to make good use of it. Life is drifting into bioinformatics presently, and I'm sure you're going to enjoy your stay with us while you're with us during this virtual internship program, and you'll learn way more than you'd actually expect. Come with an open mind, be ready to be a part of the section wholeheartedly, and I'm sure you would enjoy the internship in whole. Thank you very much. I mean, this marks the end of our meeting. We'll be saying thank you to you. We'll be saying thank you to everybody for joining. Thank you for being with us so far. We really do appreciate you. We appreciate your joining. We appreciate your commitment. And we're glad to be a part of your success story, of your research story. Thank you and do have a nice weekend and day. Thank you so much. Bye. Have a great day ahead.
Bye.
Thank you. Bye.
Hello everyone. Hello. Can anyone please give me some guidance? Hello. Is I am audible to someone? Hello. Is I am audible to someone?
30,000. Reach.